Good morning, everyone. I'm glad you can join us for this webinar. So today I'm going to discuss how we can harness cancer-mutated neoantigens for immunotherapy. I'm going first to provide a short rationale for targeting mutated cancer neoantigens. Then I will discuss our progress on better predicting neoantigens for immunotherapy. I will also provide you with a brief overview of cancer vaccines, platforms that are currently in the clinic, and specifically focus on our RNA platform. And I will present you data on our preclinical studies and how this is informing our program. So as you know, T cells are the primary mediators of cancer immunity. And as you can see on this very nice video by Alex Ritter, the green T cells are interacting very closely with the red tumor cells. And once they recognize these tumor cells, they kill the tumor cells by releasing granzyme and perforin, as you can see here with the red flash. So these T cells, are recognizing peptides that are 8 to 12 amino acid long and that are derived from the proteins that are produced by the tumor cells. For an effective immune response to be generated, there are multiple steps that need to occur. First, the T cells, they need to be primed. The dendritic cells that are antigen presenting cells have to take up cancer antigens and get activated, and then migrate to the lymph node where they can prime and activate naive T cells by presenting cancer antigens to these T cells. These activated T cells then migrate to the tumor bed when they recognize tumor cells and kill these cells. This leads to the release of more cancer antigens that can be further taken up by dendritic cells, and therefore amplifying the immune response against cancer. What we now know is that there are multiple steps at which there could be defect in this immune response, and that's why cancers are growing. The best immunosuppressive pathways that have been identified are these immune regulators, CTLA4 and PD-1, that are blocking T cell function at the tumor site. And there are now antibodies that can be used to block this checkpoint and release the T cell function, allowing the T cells to kill the tumor cells. This is very effective in many cases. One of the issues is that it is now recognized that for this checkpoint inhibition to be functional, the tumors have to already elicited a pre-existing immune response. So these tumors need to have an inflamed phenotype. What is now known is that for the checkpoint inhibition to be functional, the tumor cells have to be already infiltrated with a pre-existing immune response. Although we know now that these checkpoint inhibitors can also work at priming, it looks like most of the action is occurring at the tumor bed. So cancers with immune desert phenotypes, for which we think that there is a defect in priming, or cancer that have an immune excluded phenotype, for which we believe that there is a stromal barrier that is preventing the T cells to penetrate into the tumor. These T cells, these tumor cells do not respond to a checkpoint inhibition, and therefore there is an intense effort to identify novel strategies to be able to address this issue by immunotherapy. So one of the major limitations so far has been the poor understanding of the neoantigens, or the antigens, the cancer antigens that are recognized by these T cells. So what we know is that T cells are normally tolerized against healthy normal cells, and cancer cells look very similar to these normal cells. So the field has been focusing for many years on cancer-testis antigens or overexpressed antigens One of the issues with these antigens is that they're often poorly immunogenic because they're expressed in other tissues and there is pre-existing tolerance. And even if there is a T-cell response which is generated against these cancer-testis antigens or overexpressed antigens, these T-cells are not very effective at controlling tumor growth. So there is a second type of tumor antigens that have been long thought to be good targets, and these are cancer-specific mutations. They have the potential to be recognized as foreign or neoantigens. And they're ideal targets because they are really tumor-specific. So the major issue that the field had over the past years is that it was very difficult to identify this mutation. And it's with the advent of next-generation sequencing that we finally have been able to harness this information. So I'm showing here a figure from a seminal paper by Lawrence that was published in 2013 in Nature, where they use next-generation sequencing to reveal that there's a high accumulation of mutation in human cancer. And what you can see here on the y-axis is the number of mutations per megabase, while on the x-axis you have different indications. And there are a wide range of mutations among these different cancer types. But what was really noticeable for the cancer immunity field is that the cancers with high mutation burden are also the cancer types that are responding to immunotherapy, such as melanoma, lung, or bladder. And really, this suggests strongly that this mutation load may be associated with response to immunotherapy. So following this seminal paper, there was a wealth of studies that were done and a lot of data generated to show that these mutated neoantigens actually drive anti-tumor response. And I'm providing here a very short summary. It was shown that peptides that contain mutations can be presented on MHC class 1 as well as MHC class 2 molecules. They can be recognized as foreign or neoantigens by both CD4 and CD8 T cells. And you can see here on the left-hand side figure that the T cell response elicited against these neoantigens is stronger than the T cell response elicited against shared antigens, again suggesting that these neoantigens are recognized as foreign. In addition, it was shown that high tumor mutation burden correlates with clinical response. And this is true across different cancer indications as well as different checkpoint blockades. Here is just an example on the right-hand side with Tecentriq, which is an anti-PD-L1 in the context of bladder cancer. And you can see that the patients who are having a high mutation burden benefit from the treatment, while the patients who have low mutation burden do not. The direct evidence that neoantigens elicit an effective T-cell response come from adoptive T-cell therapy, and especially from the group of Steve Rosenberg, who showed that infusion of CD8 T-cells that are specific for the KRAS G12D mutation, which is presented in the context of HLA-C802, is inducing tumor regression in the patient. And you can see that before treatment, there was a lesion that disappeared after treatment. What is also very interesting here is to notice that these T cells do not recognize the wild type KRAS in a co-culture assay in vitro. They do not get activated. They don't produce TNF or IL-2. These T cells only recognize the mutation. So the immune system can differentiate the mutated peptide from the wild type counterpart. Again, re-emphasizing that these antigens are recognized as foreign. So following this information, there have been a lot of clinical applications that have been that have followed regarding neoantigen identification. So the first one is we can use this information as a predictive biomarker. We can use this information to select patients for immunotherapy. And that is what Merck is doing with Keytruda. They got FDA approval for the treatment of MSI high patients, which are microsatellite instability high or mismatch response deficient patients who have high mutation burden in different solid tumors. And now there is approval for Keytruda, which is the anti-PD-1 for the treatment of these tumors. More recently, there was also the approval of Keytruda for patients who have cancer with high tumor mutation burden, which is more than 10 mutations per megabase. We can also use this information for a biomarker. This allows us to generate tools such as MHC, class 1 or class 2 tetramers that enable us to follow the T-cell response. And you can see here on the right that there is a patient who, upon treatment with pembrolizumab, develop a T-cell response and against the neoantigens, and this is associated with a reduction in the tumor size. So we can track the T-cell response against a tumor. We can phenotype these T cells. We can better understand how immunotherapy is affecting the T cell response. And furthermore, we can identify novel targets for immunotherapy and novel combinations for immunotherapy. And finally, we can also develop novel therapeutics. And this is what my talk is going to focus on. So at Genentech, we are very interested in developing therapies against neoantigens, and we have two complementary approaches. We are developing a therapeutic cancer vaccine, which is directly delivering neoantigens to the patients. And this allows the activation of the endogenous immunity. There can be in vivo priming as well as expansion of neoantigen-specific T cells induced by vaccination. We are also developing adoptive T cell therapy. So we will be identifying neoantigen-specific TCRs, engineer autologous T cells with these TCRs, and then expand these T cells ex vivo and reinject them into the patients. So what is interesting is that the vaccine relies on the patient's endogenous TCR repertoire. It allows not only to expand a pre-existing response, but also to prime a de novo response. The vaccine is generating a polyclonal response, CD4 and CD8 response, and multiple antigens can be targeted. I think one of the big questions is, can we generate a sufficient number of T cells for effective tumor control? On the other hand, the adoptive T cell therapy allows to generate a very, very large number of T cells ex vivo. And we have also with this approach a unique ability to harness unique TCRs that can be found either in healthy individuals or in cancer patients, and use these TCRs to engineer T cells. We can also engineer additional modalities to get the phenotype that we are looking for these T cells. I think one of the issues is that so far we can use only one TCR per target, and this is a very complex manufacturing process, and it's likely to be limited to a small number of targets. There are also questions regarding quality and memory of this T cell response that is generated ex vivo. But one thing which is interesting is that you can potentially think that we could use vaccines to boost T cell engineered T cells. So with a vaccine, we can prime outside the tumor microenvironment, which is highly immunosuppressive. And that will allow to generate a more potent and optimal response. We can provide also the optimal adjuvant and help the immune system to focus on the protective neoantigens. With the adoptive T-cell therapy, we can generate a much larger number of T-cells, as I said before, and the big advantage here is that we literally bypass in vivo priming, and we may even able to bypass over steps of the cancer immunity cycle if we further engineer these T-cells. So a big advantage of this approach of targeting neoantigens is that all tumors are potential targets for this immunotherapy. All tumors harbor mutations. The other advantage is that neoantigens are exquisitely tumor-specific, and therefore they are optimal targets for immunotherapy, and they are likely safer. The major issue is that most tumor mutations are unique for each patient, which means that we have to develop a drug that is uniquely tailored for each individual. We have to develop an individualized manufacturing for each patient. And we also need to tailor an individualized immune monitoring. So that's a lot of hurdles. When it comes to T-cell therapy, I think what I said earlier on is that one of the big advantages is that we can harness some unique TCRs that can be found in healthy individuals or in cancer patients. And we know that there are some mutations which are driver mutations and can be found in many different patients. It seems that these driver mutations are not very immunogenic, but if we can find, by screening thousands and thousands of TCRs, if we can find a TCR with the appropriate characteristics, then we can generate a TCR library. And so from there, we know once we have determined that the patient is expressing the mutation and also has the right HLA, it's easy to match this patient to the TCR library, use the plasmid encoding for these TCRs to engineer the T cells. But as I previously said, most of the mutations are private. And in this case, what we'll have to do is identify the mutations by whole genome sequencing and the HLA type of these patients, and then use a mirror assay that has been developed by Adaptive to identify TCR that are specific for these mutations in the context of different HLA. So this is mainly focusing on pre-existing immune responses. And then we need to clone the TCRs and engineer the T cells to produce, to express these TCRs before we can expand them and then reinfuse them in the patients. So there are some vaccines that are focusing on shared neoantigens, but most of them are really targeting the private neoantigens. So we need to take a tumor biopsy from the patient, isolate the RNA and the DNA, as well as normal cells, DNA from normal cells, to identify the mutations that are expressed. We need then to predict which of these mutations are more likely to be immunogenic, rank these mutations, and then incorporate them into a vaccine. So what are the components that are critical for individualized cancer vaccines? Well, we need a potent platform. We need a platform which is good at eliciting CD8 T-cell responses. But we also need a platform which is simple and amenable to individualized tailoring. We also need to have a good neoantigen selection algorithm. And only if we have both a potent platform and an effective neoantigen selection algorithm will the vaccine induce immunity. And then there is something which is also very important to think about is the end-to-end manufacturing. We need to think about turnaround time. The patients do not have the luxury of time. they often have metastatic cancer and they need a vaccine as soon as possible. It's also taking time for the vaccine to generate an immune response. So if we think about all of this, it's very important to identify a vaccine that can be generated quickly, but also that is potent at inducing a very rapid T-cell response. So I'm going to focus over the next slide on antigen prediction. So in order for these antigens to be presented to T cells, first the proteins have to be degraded by the proteasome into small peptides. These peptides are then transported through the TAP transporter into the ER, where they are loaded onto MHC class I. And the complex MHC class I peptide is then transported to the surface for presentation to the T cells. And the T cells next have to recognize this complex as foreign to be activated. So this is a multi-step process, and only a small fraction of the mutations are actually making it through this process. So only a small fraction of these mutations are immunogenic. So there have been a lot of prediction algorithms that have been developed over the past years to enrich for immunogenic mutations. So what we are looking at in general, what are the criteria for selection? We need to think about tumor expression. You know, what is the level of expression of the gene? Is there clonality? And what is the variant allele frequency? Presentation is an absolute. We need to validate that these mutated peptides have high binding affinity for MHC class I. And then immunogenicity is something which is also important. And a lot of different groups have been considering looking at the relative affinity of the peptides in comparison to the wild type counterpart. So there have been some studies that have been published showing that indeed we can identify or predict effectively immunogenic mutations. But if you look at these 3 different studies that have been using different platforms, only about a quarter of the mutations that were predicted to be immunogenic are actually immunogenic. So we need to improve this prediction algorithm. And we can do it two ways. First, we need to improve our ability to predict presentation on MHC class I. We need to generate more mass spec data and apply machine learning on this database of peptides that have been identified by mass spectrometry. The big advantage of this approach is that not only we are looking at binding affinity, but also these peptides are resulting from processing and transport into the ER before being loaded onto MHC class I. So we are looking at more steps than just binding. The limitations is that mass spectrometry, although it's really improving, is still relatively poorly sensitive. And it's a binary outcome. Either we see a peptide bound or we don't see a peptide bound. And there are also some bias that can be associated with mass spec. Not all peptides are flying as well. And there are some issues also with immunoprecipitating of these complexes. We're often using pan antibodies, but these antibodies are not recognizing equally all different alleles of MHC class I. In regard to immunogenicity, not only the peptide has to be presented to the T cells, the peptide has also to be recognized by these T cells as foreign. So we need to generate a larger immunogenicity database, and that will come from preclinical model, as we have already done, but also from all the clinical studies that are being done looking at pre-existing immune response as well as vaccine-induced responses. I think it has been very important for the field to really do an in-depth analysis of the T-cell response following vaccination, so we can better understand the determinants that are leading to immunogenicity of these peptides. So what we have done at Genentech and what other groups have also done is generating a lot of mass spec data We have over 600,000 presentation data points. We use machine learning, and that includes different features that are associated with presentation, such as the amino acid sequence of the peptide that is bound to MHC class 1. We have also looked at the flanking regions around this peptide, as well as the contact residues of MHC1. And by generating all this data, we have been really able to improve our prediction algorithm. And here, if we compare it to the next MHC class 1, that is a method that is considered the state-of-the-art method and that is available publicly, you can see that our GAI method is doing better in terms of area under the curve. For example, our GAI method is 0.75, while net MHC is only 0.52. And with an area under the curve, we are really trying to aim to 1. So we are also looking at the determinants of immunogenicity. We know that the absolute affinity of the peptide into the MHC class 1 group is important. But not only the peptide has to bind MHC class 1, it has to be recognized as non-self. And so, as I mentioned before, relative affinity is important. If the wild-type peptide is not predicted to bind, for example, then only the mutant peptide is presented. And that suggests that the whole sequence has never been seen by the immune system and is more likely to be immunogenic. What we have also shown is that mutations, the position of the mutation is important. So if the mutation that you can see here on the lower left-hand side in yellow is in the middle of the peptide, you can see here is the MHC class 1 groove with the peptide bound to MHC class 1. And so if the mutation is in the middle of the peptide facing up towards the TCR, it's more likely to be recognized as foreign and immunogenic. While if the mutation is hidden into the MHC class 1 groove and not seen by the TCR, by the T cells, it's less likely to be immunogenic. There are also some studies that have suggested that similarity to microbial epitope may be important for these peptides to be recognized as foreign. And there are also studies suggesting that the TCR peptide affinity is important, and that the bulkiness as well as the hydrophobicity of the residues that are in contact with the TCR play an important role in leading to immunogenicity. So what we decided to do in my group is to identify immunogenic neoantigens in our mouse tumor models. And we have predicted MHC class 1 neoepitope just by looking at expression and binding using an HMHC score of less than 500 nanomolar, and in 4 different tumor models. We identified about 400 of predicted immunogenic mutations. And then what we did is we immunized mice using long peptides plus strong adjuvant poly-IC and anti-CD4 T, and measured the T-cell response against these antigens. And what we found is that only about 10% of the predicted mutations are actually eliciting an immune response. And this is very similar to what has been seen in the clinic in terms of frequency. Then we looked at these peptides. And what we found is that we have 2 types of mutations. We have mutations that are at anchor residues. They show an increase in affinity over the wild-type counterpart. And we have also mutations which are at non-anchor residues. And for these mutations, the IC50, the binding affinity for the wild-type peptide, is equivalent to the one of the mutated peptide. What is interesting to see here is that we have about the same frequency for both types of mutations. About 10% of these predicted immunogenic mutations are actually eliciting an immune response. But we have way more mutations that are at non-anchor residues simply because most of them are only 2, maybe 3 out of 10 amino acids, which are actually anchor residues. So mutations that are at non-anchor residues also play an important role. So then we wanted to further understand how these mutations behave. And so we looked at immunogenicity of these mutations. What is the strength of the T-cell response measured by interferon gamma against these mutations? And we didn't see any difference. All the mutations at anchor or non-anchor positions elicit very comparable immune responses. What we observed is that there is somewhat of an enrichment in the middle of the peptide at position 4 for the non-anchor mutations. And as I told you before, we had already preliminary data suggesting that if the mutation is in the middle, facing up towards the TCR, it's more likely to be recognized as immunogenic. And we also confirmed these results using a published dataset of human mutated peptides. And you can see here that there is an enrichment for mutations that are position 5 when it comes to human peptides. Then we wanted to understand how we can use the predictive value of the binding affinity in the context of mutations that are at anchor residues or at non-anchor residues. And what we found is that if the mutations are at anchor residues, what is important to look at is the relative affinity in comparison to the wild-type counterpart. So you can see on the upper right that we can distinguish for human and mouse peptides that when we look at relative affinity, we can better predict which peptides are immunogenic. If we are looking at absolute affinity, there is no difference between the immunogenic and the non-immunogenic peptides. When it comes to non-anchor mutations, relative affinity, as expected, doesn't play a role. But what is important in this case is the absolute affinity of the peptide for MHC class I. The higher the affinity, the more likely the peptides are immunogenic. And so we think that including the anchor context is very important when it comes to ranking neoantigens based on binding affinity. So we have been focusing mainly on single amino acid changes because they are the most abundant. But I think it will be also interesting to look at other types of mutations, such as insertions, deletions, or fusions, that may be more disruptive, more different from the self, and they may be more likely immunogenic. There are also other categories of neoantigens that we should look at, potential post-translational modifications that could alter recognition of the TCR for these peptides. And there is also quite a lot of work going on at looking at the potential of endogenous retroviruses or dark matter. As these retroviruses are dysregulated, their expression is dysregulated in cancer, it may be that they could behave as neoantigens. There are also other factors that we need to take into account, especially tumor expression. What is the expression of the mutated proteins? What is the allele of expression by tumor cells? We know that one of the major issues we are going to face is that MHC class I expression is often downregulated in tumors. Clonality is also very important. The goal is to target tumors that are expressed by most of the tumor cells to avoid escape. And finally, something that the field is starting to look at is the role of the CD4 T cells. There is more and more data suggesting that they play an important role, although it's not fully clear what they are doing. But their prediction algorithm is relatively poor. Although, on the other hand, what has been interesting is that the vaccines appear to be better at eliciting CD4 T cell responses than CD8 T cell responses. So we may not need to have a very good prediction algorithm for the CD4 T cells. This has to be seen in the future. So I'm going to switch gear now and discuss vaccine platforms. So there have been quite a few studies that were published over the past few years using different vaccine platforms, a DC-based vaccine, an RNA-based vaccine, as well as peptide-based vaccines. What is interesting with these studies is that the good news is that they show no severe adverse events. The other interesting thing is that the majority of the patients showed an enhanced T-cell response in the blood. They develop both CD4 and CD8 T-cell responses. What is also important to note, as I said before, is that only a fraction of the selected mutations were immunogenic. And I think the major question here is it that the platform is not optimal? Is there some type of immunodominance going on? This is a phenomenon that is very well known in the infection field as well as in the tumor field. Some peptides or some mutations or some seem to be dominating the immune response. Or is it that our prediction algorithm is not fully optimal? It's probably a little bit of both, but further studies are needed to better understand the contribution of the platform and the algorithm. These studies, the Keskin studies in 2019, showed that the vaccine induced expansion of the T cells at the tumor site. Either the T cells migrated to the tumor site, the activated T cells migrated to the tumor site, or they were local activation. But that's interesting to see that these T cells can reach the tumor. When it comes to clinical response, I think, this is the data that are really missing. There are some anecdotal suggestions that these vaccines can be effective, but many of the patients that were treated had no or limited metastatic disease. The reason is that it takes time to make this vaccine currently, and so the patients that were chosen had usually low or limited tumor burden. So the jury is still out there when it comes to efficacy. There are a lot of different platforms currently in the clinic. This is not an exhaustive list of companies, but this is a competitive field. What is interesting is that there are a lot of different vaccine platforms. RNA vaccine, DNA vaccine. There are also some companies choosing vectors, so viral vectors as well as bacterial vectors, and companies have also decided to go forward with an heterologous prime boost, which has been shown to be very effective, but it's also very challenging when it comes to manufacturing two different vaccine platforms for an individualized approach. So we are eagerly waiting for the results of these clinical trials. So Genentech, in partnership with BioNTech, has a program, a cancer vaccine program. And what we chose is an RNA-based vaccine. The reason why we like the RNA-based vaccine is that it's easy to make an RNA. And the RNA has the two functions that are required for a good vaccine, a good platform. So the single-stranded RNA is a TLR7 and 8 agonist, so it provides the innate stimulation, which is necessary to activate dendritic cells and promote T-cell response. And the RNA is also a messenger RNA that is expressing the neoantigens. Up to 10 neoantigens can be expressed in this cassette. The RNA has been engineered such that we have signal sequences, at the N-terminus and the C-terminus of the polypeptide to increase presentation on MHC class 1 and MHC class 2. And we have also 5' UTRs and 3' UTRs that have been engineered to improve expression. This RNA is encapsulated into a liposome that has been optimized for delivery IV and targeting of the spleen. In the spleen, the RNA lipoplex is taken up by macrophages and dendritic cells. As I said earlier, the RNA is stimulating TLR7 and 8. This leads to the production of pro-inflammatory cytokines as well as co-stimulatory molecules that are required for priming of naive T cells. The RNA also somehow is translocated into the cytosol, where it's translated into a polypeptide that is degraded into peptides that are either loaded on MHC class 1 for presentation to CD8 T cells, or that are loaded on MHC class 2 for presentation to CD4 T cells. So the RNA lipoplex has been evaluated in advanced solid malignancies, and the data have been presented at AACR last month. We have tested the vaccine in phase 1A as monotherapy. It is overall very well tolerated, and the majority of the patients, 86% of the patients that have been evaluated, showed an enhanced T cell response in the blood after vaccination against neoantigens that were found, that were incorporated into the vaccine. We identified both CD8 and CD4 T cells. We also have shown the data of our phase 1B that is in combination with atezolizumab. And again, the combination is well tolerated, and the majority of the patients that were evaluated showed an enhanced T cell response in the blood. We have also preliminary evidence of expansion of vaccine-induced T cells at the tumor site, again, suggesting that these T cells can migrate to the tumor. So these studies were very interesting, but not really aimed for looking at efficacy. So we have now 2 ongoing randomized phase 2 studies to better understand the impact of the vaccination on anti-tumor efficacy in combination with checkpoint inhibitors. We have a study in metastatic melanoma patients together with, in combination with pembrolizumab, a PD-1 antibody. It's for the treatment of first-line. It's for first-line treatment of patients. We also have a study in combination with atezo in an adjuvant setting in patients with non-small cell lung cancer. I think one interesting advantage of this study is really to understand if in patients with low tumor burden and also maybe where there is less immunosuppressive environment, can the vaccine in combination with atezo be more effective? I'm going now to switch to what we have learned from our preclinical studies. So our goal was to mimic what we do in the clinic. So we decided to design 2 RNA decatopes, each encoding for 10 mutated antigens that we had identified in the MC38 colorectal model. And so we immunized mice in order to better understand if these T cells are migrating to the tumor, what is their function at the tumor site, and if we could see any correlation between tumor size and protection, as well as studying combination studies. We also looked at the requirement for efficacy. And so what you can see here is that these 2 decatopes elicit a T cell response against multiple antigens. Each color indicates that a different antigen is recognized by these T cells. And they, both decatopes elicit a T cell response overall that is very similar in terms of magnitude as seen by interferon gamma production. So the first question we wanted to ask is, Can we induce with the RNA lipoplex a functional T-cell response that gets to the tumor? And yes, indeed, after vaccination, we observe that there is an increase in the number of T-cells that are infiltrating the tumor. And you can see here that in addition, in the middle here in the panel, we see that there is an enrichment for neoantigen specific T-cells at the tumor site in comparison to the spleen, suggesting that these T-cells do preferentially migrate or proliferate at the tumor site where they recognize antigen. We also look at the function of these T-cells, and they produce at the tumor site quite a lot of interferon gamma, and this is dose dependent. The higher the dose of vaccine, the higher the production of activated T-cells at the tumor site. Next, we wanted to ask if this T-cell response is functional and how often we needed to vaccinate. And here is again the MC38 model. We immunize tumor-bearing mice with the vaccine once, twice, or 3 times, or even 4 times. So we are vaccinating every week. And what you can see is that the vaccine is effective at controlling tumor growth, but what is also interesting is that it seems that we need to continuously vaccinate the mice for this T-cell response to continue controlling the tumor. If we vaccinate only twice, we see quite a few tumors which are actually growing again after vaccination, while vaccination 4 times is leading to a more profound response. So we wanted to further understand what was going on. And so what we did is we looked at the T-cell response after immunization at day 5, day 12, and day 19. And what you can see on the left-hand side is that there is a very nice T-cell response, polyclonal T-cell response against different neoantigens that is generated at the tumor site. And we see a very similar magnitude of T-cell response up to 19 days post-vaccination. What is more interesting is that when we look at functionality of these T-cells, what we observe is that these T-cells lose functionality very quickly after vaccination, suggesting that they are getting exhausted. And if we revaccinate these mice, what we observe is that there is an increase in proliferation of these T-cells and functionality of these T-cells. So continued vaccination is needed to maintain a pool of proliferating and functional CD8 T-cells. We wanted to also see if there is a correlation between the tumor size and response. So we vaccinated mice at different times after implanting the tumors in mice. And what we found is that the earlier we start vaccinated and the more profound is the T-cell response. When the tumors are too big at day 18, they barely respond to vaccination. And that could be for 2 reasons. It could be that the T-cell tumor cell ratio is critical. There are too many tumor cells at day 18 for the T-cells to effectively control tumor growth. But it also could be that the tumor microenvironment is more immunosuppressive in this larger and more established tumors, and that require combination with checkpoint inhibitors. So I think this is a good argument for testing a vaccine in an adjuvant setting and also in combination with checkpoint inhibition. So that's what we did. We tested our vaccine with anti-PD-L1, and as you can see here, vaccination alone is leading to tumor regression. But when it is combined with anti-PD-L1, we do observe a more profound response, suggesting that these T-cells are more effective and that combination is needed. So I just want to finish here by presenting some very preliminary data, which I think are really interesting. We wanted to better understand, what is beyond immunogenicity? Is immunogenicity sufficient for tumor killing? So in the context of the vaccine, we can elicit a large number of T cells with a wide breadth. Kinetics is controlled, and we can also control the quality of the T cells that are generated depending on the route of immunization or the adjuvant that are used for vaccination. But it's different from T cell function in established tumors. How is tumor killing occurring? We need tumor recognition, so their antigen clonality and expression and presentation is important. But we need also to have tumors that are sensitive to the killing. And in addition, we need to have T cell survival and T cell sustained function in this tumor immunosuppressive environment. So we went back and decided to look at our two decatopes. All the studies that we done previously combined decatope 1 and decatope 2 for immunization. So this time we decided to see, to immunize only with decatope 1 or only with decatope 2. And what you can see on this slide is that decatope 1 is not at all effective at controlling tumor growth. It's barely different from the no treatment or control vaccine. While it seems that it's decatope 2 that is really important at eliciting a protective immune response. This was quite surprising to us. If you look at the two decatopes, as I said earlier, they elicit a very similar T-cell response in terms of magnitude in healthy mice. They elicit a lot of different T-cell responses. Decatope 1 seems to be even eliciting a broader breadth of responses. We looked if the T cells were getting to the tumor, and with both decatopes, we see an increase in T cells at the tumor site. So there is no issue with T cell homing to the tumor. If you look at the total number of T cells, it's very similar with both decatopes. And if you look at the T cell specific, tumor specific T cell response using tetramers, you have also very similar accumulation of T cells at the tumor site. Then we decided to do a prophylactic vaccination. So first immunize the mice and then challenge them with MC38 tumor cells. And we had the surprise to find that decatope 1 is very effective at controlling tumors when the decatope vaccination occurs before we challenge the mice with the tumors. And it's as good as decatope 2. So which means that decatope 1 can induce T cells that recognize and kill tumor cells. So why don't they control tumor growth in therapeutic settings? We don't have a good answer yet, but this is, I think, very important to understand. Some antigens are providing protection in prophylactic settings only, while others are providing protection in both prophylactic and therapeutic settings. And it's very important for us to understand what are the determinants of this difference. Can we identify these antigens that are eliciting a T-cell response which is effective in therapeutic settings? We are trying to understand if these differences is the reflect of different tumor neoantigen properties, such as clonality, expression, or presentation, or is it related to the fact that there is a pre-existing immune response against some of these antigens? Maybe if there is a pre-existing immune response, vaccinating is going to elicit a faster, stronger response, while if the vaccine has to elicit a de novo response. Is there also something specific with the tumor microenvironment? There are some studies that are suggesting that very high affinity T cells are actually getting exhausted faster than T cells with lower affinity. So maybe there is a TCR affinity sweet spot where the affinity is sufficient for killing the tumor cells, but not too strong so that these T cells get exhausted. And then the next question is, how can we overcome this lack of efficacy that we have observed with some neoantigens? Can we use combination therapy? or do we have to deliver a very large number of T cells through adoptive T cell therapy in order to overcome this issue? So I just want to finish by saying that I hope I convinced you today that neoantigen-specific cancer vaccination can potentially be effective at controlling tumor growth. We need to identify the right vaccine platforms that are potent in terms of immunogenicity, but also that can be tailored for an individualized patient. It's also very important to improve our neoantigen selection because only a small fraction of these mutations are actually immunogenic, and we need to identify antigens that can elicit an effective T cell response. And the characterization of these neoantigens is critical for this identification. Finally, I want to add that identifying the right patient population is important. Not only the tumor have to have high tumor burden, but we also need to consider the tumor burden itself. And it's possible that by testing this vaccine in adjuvant settings where the tumor burden is lower, where there may be less immunosuppressive environment and more time for the vaccine to be generated and for the T cell response to be mounted in each individual, will provide the key for success. I also want to add that combination therapy will be critical. We need to identify partners that can improve the magnitude of the T cell response, help further increase the migration of the T cells to the tumor, and improve the sustained functions of the T cells that are induced by the vaccine at the tumor site for success. And with that, I just want to thank everybody in the cancer vaccine team, both at Genentech and at BioNTech. It has been a real honor working with all of these people on this exciting project, and I want to thank you for listening.